Well, I started recording early again, as usual. So if you're watching this video, obviously there's a lot. We've got about 30 minutes before the lecture actually starts. So your first word for attendance is going to be wheelchair. That's your first word for attendance. And I guess you can fast forward until you see the screen change, and then I can't guarantee that's when the lecture starts, but it might be. So when you see the screen change, maybe quit fast forwarding and start listening to what might be being said during the video. And that's it. So, yeah. morning, Kinder.
Good morning.
Usually people online don't get extra credit, so go ahead and give you a word. That way you'll have extra words, so you will get at least a little bit of extra credit. And that first word is going to be door.
Thank you.
You're welcome. Good morning, Hannah and Cook. I guess I'll do another extra credit word since you guys are 13 minutes early. Let's see. And that word is going to be smoking. Like if you were smoking a brisket or smoking a cigarette. Smoking is the first word, and it is extra credit. Well, not the first. You guys missed the first word, but that's okay. It's extra credit. Speaking of which, as always, if you send me the words and I don't give you extra credit, remind me that those were extra credit words. Or, as sometimes is the case, sometimes people will get the extra credit words, but then miss some of the regular credit words, and then they end up just having full credit instead of extra credit, but that's still better than nothing. Morning. Good morning, Burton.
Good morning.
Good morning, Durgan. Morning.
You do this after class.
Yeah, because I'll have to download something.
All right, you're good.
I'll download it now. That way, after class, I can just fill it out. We'll be ready to go.
Did you say if we went ahead and finished the online lab, we didn't have to come to class?
That'll be one of my announcements. As a matter of fact, when I'm done with my announcements, if I didn't say anything about that, please remind me.
All right, I got you.
I'm going to talk about that, attendance words, the exam. Yeah, that's the most important stuff. Excuse me. Good morning, Fisher and Hughes. So online so far, we have Kinder. We have Hannah. We have Cook. We have Burton. Fisher. Hughes. Durgan. That's it online for now. All right. Actually, do you have a pen? Yeah. Might as well do it now. At least get it started. There you go.
Thank
Morning. Good morning, Cooper. Let's see, got a little bit more than 3 minutes. So to remind you guys in person, today's extra credit words are not extra credit. They're proof that you were paying attention to the lecture. So make sure you send the words in. That's basically what I'm saying. And that you're paying attention. Morning. All right, we've got a little bit more than 2 minutes, so I'll go ahead and start attendance. The first word, well, it's not 8 yet, so this one actually is extra credit, will be button. So the people who've been online for a while know it's not the first extra credit word, but most recent extra credit word is button. Anyway, let's do some attendance here. Morning. So, Fisher, again, if you're online and I say you're not here, just ignore it. It doesn't matter. Again, just send me the words afterwards and you'll be counted. But anyway, Fisher is online. Zach is not here. Canterbury is here in person. Jones, not here. Kinder's online. Burton's online. Tate is here. Durgan's online. Cook is online. Galloway is not here yet. Hughes is online. Madeline is not here yet. Larissa's not here yet. Hannah is online. Kennedy's not here yet. Talbert's not here yet. Julia is online. Melton, both are here in person. Reinhardt is here in person. Crowder is here in person. Like I said, Melton is here in person. Hunter is here in person. Cooper's online. Lewis is here in person. Mullins is here in person. Andrea is not here. Kincaid is not here. Abigail is here. Kennedy is here. Anybody I miss in person? Well, it's 8. I'm going to hold off till 8:01. But I'll do some announcements. I guess I should do the least important announcements first, since hopefully other people show up. So for lab, it's a really, really easy lab. It's in person. It doesn't require teamwork. So I'm going to be a lot less stringent as I have been. So if you've sent me the lab and you think you're done, I'll take your word for it. I'm going to grade it, but if you say you're done and you don't think you need to come to lab, then don't come to lab if you've done it. Normally, I'd be like, I need to check it first and make sure that you've done it right. But again, it's an easy lab. Doesn't require teamwork, so whatever. Likewise, well, you still need to make sure you do the pre-lab. If you do plan to come to lab, make sure you still need to do the pre-lab first. But if you haven't done it yet, today, just don't come to lab today. Come on Wednesday, because I'm not going to have time between now and lab time to do that. So Larissa's here, and Zach is here. Anyway, the other thing about lab 2, like I said, is trying to have a little bit more fun now that we're over the halfway point of the semester. So if you do want to come to lab, you can either come today at 11 as usual, Wednesday at 11 as usual, or excuse me, 10 as usual, Wednesday 10 as usual, or I'll be at the Roca's today at 11, and I'll have my computer. I don't plan on doing any lab work, but if somebody wants to come and needs help, I'll be there to do it. That and drink margaritas. So, any questions about lab? All right, what are some other ones? The exam, thanks to my stupid fat fingers, I had a typo and made a big mistake. And the exam was, I wrote that it didn't close until Tuesday, which is supposed to be Friday. But anyway, it's still open because of that. If you've already taken it, obviously it doesn't matter for you. But if you haven't taken it. It closes tomorrow. One of you I saw sent an email. I haven't had a chance to read it. I just saw the subject line. Anyway, and then they said they had an issue with the exam. So if you're trying to take the exam and it's not working, let me know. Like, meet me online after this class or come to my office, either way, and we'll figure it out. I think that's it. And then once the exam is closed, which is tomorrow, then everybody will get your grades, right? Until then, I don't even want anybody knowing what the grade is, much less which questions they got right and which questions they got wrong. So any questions about the exam or the lab for that matter? All right. Independent work, you should have 90 points by now. This is the last week of independent work. It's going to really suck for me because, as usual, people like to wait till the last minute to turn it in. So let me just say, that's plenty of time. Never mind. Yeah. So finish up your independent work this week. Yes, sir.
We can't turn any independent work in after this week.
Great question. It'll be like every other thing as far as late work is concerned. Officially, I'm not taking any independent work after this week. Unofficially, send it on in. It's just going to be at the bottom of my priority list. You know what I mean? Like, you'll send the email and it'll just sit there in my email. As new stuff comes in, I'm grading new stuff. And then once I'm caught up with all my new stuff, then I'll go back and grade stuff that's past due. Does that make sense?
Yeah.
Yeah. And it's early in the semester. I did that on purpose, right? So, again, officially, it's too late. But unofficially, I'm going to do my best to get that stuff graded. That'll be all the extra credit will be applied at the end. Oh, that's a good question too. Yeah. So yes, you can still send in papers because they will be extra credit. But again, I'm going to put those at the bottom of my priority list. Written work, just because it takes, as you guys see how I grade. I go through it like a fine, like very, very picky usually. So it takes me a lot of time to do it. So yes, you can still turn stuff in after this week. It's just, again, officially, it's too late. Unofficially, I do my best to grade it. Great questions. Anything else? All right, what else? So independent work, lab, exams. Next month, there's going to be a Friday where I can't come in, and we're going to have an online class. I think it's the 26th. I'll tell you for sure when I know for sure. What else? I think that's it. Yeah, I think that's it. If I think of anything else, I'll let you know. Yeah, let's get started. The first word. Oh, yeah. So today's, remember, the words for attendance today are for attendance. They're not for extra credit. So make sure you're writing them down and send them at the end. And on that note, let me pull up the PowerPoint. And PowerPoint will be the first official word for attendance, for participation. There we go. Also, again, we're halfway through the semester. Monday at 8 a.m. So I really am impressed with all of you for being here and the ones who are online. So everyone's going to get a little bit of extra credit just for being here online or in person. Only people who aren't getting extra credit today will be anybody who watches the video instead. But thank you for being here. I know it's tough on a Monday. So anyway, this is where we left off. Sex-linked genes. This is the last thing we were talking about when we're talking about genetics, right? Find these sex-linked genes. It's the last discussion. Yeah, let's just go through this quickly. There won't be that many questions about sex-linked genes on the exam. And what is a sex-linked gene? It's just like it sounds. It's a gene that you find on one of the sex chromosomes, right? So if it's a gene found on your X chromosome, that's a sex-linked gene. A gene found on the Y chromosome, that's a sex-linked gene. Most of them are found on the X chromosome, which is on the left, right? Whoops. And not the Y. Anybody want to guess why that is? Why do you think there's more genes on that one on the left than the one on the right? But if you don't overthink it, it's really easy to figure out. Just look at it. What do you think? That's it. Yeah, the X chromosome is just bigger, right? It's just like I've used this analogy. I'm going to keep using it. You can think of your chromosomes as books, right? Books that have instructions on how to build proteins. And they're not all the same size, right? They go from larger to small. Like, so chromosome number 1 is really big. Chromosome number 22 is really small. But yeah, the bigger they are, the more genes there are, generally speaking, right? Matter of fact, that would be a good independent work topic too. You can test me on that because I'm actually not sure. That's a hypothesis. Now I'm curious. If my hypothesis is correct, then that would mean chromosome number 1 has more genes than chromosome number 2, which has more genes than chromosome number 3, so on and so forth. So you can look that up for independent work if you want. I'm not sure because as you're going to learn, somebody reminded me of something. Thank you. So yeah, I can't fix the zoomed in part. Unfortunately, it's just messed up, but I can show the PowerPoint. So thank you for whoever was online reminding me of that. Come on. There we go. Anyway, yes, you could look that up for independent work. Not that it's important. You know, generally speaking, I think independent work, the topics are usually not important. If they were important, I would teach them, but it's just little trivial things you can figure out. Anyway, any questions about what a sex-linked gene is or where they're found? All right. Some human conditions, including red-green color blindness, hemophilia, and a type of muscular dystrophy, all result from a sex-linked recessive alleles. Now, I'm going to put it next to this because I'm not going to ask you. This is not a health class, so I'm not going to ask you. There will be no question on the exam that says, well, red-green color blindness is an example of what, right? So you don't need to know anything about red-green color blindness or hemophilia or any of the other stuff. I'm going to talk about it because it's a good example, but you don't necessarily need to know about it. If I were to give you any questions about red-green color blindness or any of this other stuff, for the question, I would give you the pertinent information, right? So it's not like you need to memorize any of this. Everything I would tell you, all the pertinent information, this stuff right here, I would give you in the question. Anyway, red-green color blindness is a very common sex-linked disorder caused by malfunction of light-sensitive cells in the eyes. It's usually males that are affected. We'll talk about why here in a second. Heterozygous females do have some defects, so they're like slightly colorblind, but not completely. And the next slide is going to show a test for it. So, for example, can anybody not see the number in that on that slide? Of course, you're at an angle, so it might not. I could see. You can see it. So nobody. There's nobody. Everybody can see it. Everybody can see the number 7. What about the number 3 next to it? No, I'm just kidding. It's just a 7. You're good. So no one here is colorblind, at least not the red-green color blindness. You passed that test. Anyway, because they're located on the sex chromosomes, sex-linked genes exhibit unusual inheritance patterns. Because again, it's sex-linked genes, right? So it's either on the X chromosome or on the Y, and those things also determine what sex you are. So because of that, your sex might have changed your chances of getting some of these things. So let's talk about them. Also, another thing I want to mention for independent work is these are just some examples, right? So I'm giving you some examples, but if you want, you can look up some other examples. What are some other examples of sex-linked genes? And also, here we were just talking about humans. What about other species? Do they have sex-linked genes? Anyway, so here's an example. Here we have a Punnett square where we have obviously a male and a female. The female is up top left. She's completely normal. You can see she has those two. This is a recessive disorder, remember? So to have it, 100% of your alleles have to be recessive, right? Just as usual, 100% of your alleles have to be recessive to have the recessive condition. So keep that in mind. Anyway, that female is completely normal. She's homozygous dominant, which means not only is she normal, but she's not a carrier either. That male is affected. He is colorblind. And again, notice I didn't say you had to have 2 recessive alleles. I changed the way I said it this time just to say 100% of your alleles have to be recessive. Does that make sense? Because prior to this, especially in lab, we would have to say, to have the recessive phenotype, you have to have 2 recessive alleles. But a better, more accurate way of saying it, now that we know about sex-linked genes, is 100% of your alleles have to be recessive. So for that male, since he only has 1 X chromosome, 100% of his alleles are recessive, right? He only has the 1 recessive allele, but again, that's 100% for the male because the male only has 1 X chromosome. So anyway, you do the Punnett square, and this is the kind of question you would maybe get on the exam, but all the pertinent information, I would give you that. So you would know it's a recessive allele, a recessive condition, excuse me. You would know it's on the X chromosome as opposed to the Y. All that pertinent information would be given to you. You would just do a Punnett square, and then the question would say, what are the chances of this person mating with this person and having a normal male? And you can see here, the chance of having a normal male are 100%. Well, excuse me, 50%, right? It's 50% chance of having a normal male and 50% chance of having a carrier female. So all that makes sense? Again, you would just do the Punnett square and come up with that by yourself. There's 2 different genotypes. There's 2 different phenotypes. So if we were thinking about this in the context of lab, right, when you do the genotype frequency and the phenotype frequency, you could say 2 to 2 for the genotype frequency. You could also say 2 to 2 for the phenotype frequency. Anyway, any questions about that example? All right, here's another example. So now we have a carrier female who is mating with a completely normal male, and then you can see the chances are a little bit different. So here we have a completely normal female, right? Here we have a carrier female. Here we have a completely normal male. And here we have an affected male. So that phenotype ratio is like anything you've ever seen, or unlike anything you've ever seen in lab, right? So the phenotype ratio in this case is 4. Or excuse me, there's like 1 1 to 1 to 1 to 1, right? There's 4 different phenotypes. Likewise, there's 4 different genotypes. But again, for the exam, you might get that question with all the pertinent information. And I would just say, this person made it with that person. What are the chances of the child being, whichever I might ask about? A normal female, a normal male, a carrier female. A male can't be a carrier, right? Because if it has the one of the recessive alleles, then they're going to be colorblind. The next word for attendance, and for those of you who came in late, again, these are not extra credit words today. These are just the words to get your attendance, is carrier. So make sure you send those words sometime before 9 a.m. All right, so any questions about this example? I think we have one more example, and I'm going to go through this quicker. Yeah, one more example. So here we have a carrier female who's mating with a colorblind male. And then again, you can see the possible outcomes. We have a 25% chance of having a carrier female as a child, 25% chance of having a normal male as a child, 25% chance of having a affected female as a child, and 25% chance of having an affected male as a child, or as an option, right? And then, or you could also say there's a 50% chance of having a child who's colorblind, right? If you don't get specific and say male or female, there's a 50% chance of that. There's a 25% chance of having a completely normal child. There's a 75% chance of having a child who's either a carrier or who's affected. Does all that make sense? All right. And if this is confusing you a lot, don't let it stress you. Do your best. Try to learn it. Let me help you if you need it. But if there's any questions about this on the exam, there's not going to be a lot. So don't let it stress you if this is the one thing that you just can't get. Hemophilia is another example. The old textbook included it, so did I. I think the new one does too. I'm not going to ask you any questions about hemophilia, but I do like that topic because this is one of those situations where biology affected history. If any of you history buffs, you might be very familiar with this. Back in the day in Europe, that's how they used to strengthen ties amongst countries, was they would be marrying, right? They would marry, this royalty would marry that royalty. And a lot of times, to keep things pure, there would be a lot of inbreeding. So because of that, what was usually a pretty rare thing, which is hemophilia, was a little bit less rare amongst the royal families because, well, they were inbreeding, right? So that's not a good thing. Anyway, you can read about it. I'm not going to ask you any questions about it, but it really is a good example. You can see the Russian Revolution. I wouldn't say it's because of hemophilia, but it had a little bit to do with it. This child, Alexis, had it. Because of that, they had somebody named Rasputin that was living in the royal palace, and that person was widely untrusted by a lot of the Russian people. And because of that, that had a lot to do with the revolution. But you can look into it if you want for independent work. Another great topic for independent work if you're into history. What are some other examples of how biology has affected history? There's a great book. What's it called? Guns, Germs, and Steel, I think is what it's called. And it's not just about biology, but it has obviously germs has to do with it. But it's about how Europe became so powerful, and a lot of it did have to do with biology, because unfortunately Europeans spread disease when they went from one place to the other. Anyway, any questions? Just again, hemophilia is just an example. Certainly not going to ask you any questions about that on the exam. Yeah, and that's it for that one. So let's go to the next chapter. I'm not going to ask you, if you're studying the study guide or studying the PowerPoint, just know that this was left over from the last textbook. So I'm not going to ask you any questions about that. And I'm not even going to go over it. We'll save our time and jump into the next chapter. And chapter will be the next word for attendance, since we're going to start the next chapter. This is probably one of the more relevant chapters, I think, right now. Of course, the pandemic is largely behind us, COVID-19, but the controversy is still there. There's still people whining and moaning about the vaccine and things like that. So this chapter is going to teach you the basics of what you need to understand how viruses work, how vaccines work, how mRNA vaccines work, all that stuff. Because I love it online. But somebody said something about the vaccine, and I just said something. Oh yeah, they said, I bet you regret taking that. And I was like, no, actually I don't. I'm okay. And then they said something like, oh, it's going to alter your DNA forever. And I know that's a bunch of BS because that's what my thesis work was mRNA. So I asked him, I said, oh, well, how's that work? You know, trying to see how do they know that it's gene therapy is what he claimed. And he said to do my own research. So I just sort of told him that that's was my thesis work was in mRNA. So I have done my own research. And mRNA does not affect your DNA. And you're going to learn how all that works here right now. Anyway, so that's basically this discussion. Why molecular biology matters? Well, that's the most relevant thing for us right now, I would say. This big debate between vaccines, should vaccines be mandated, stuff like that. And for you, I'm not going to tell you, that's an opinion, right? So your opinion, what you vote for, whether you think they should be mandated or not, or what the exception should be, that's your decision. I'm not trying to influence you. But I want to give you the information so you can make an informed decision, unlike this dude who told me to do my own research because he didn't know how mRNA was supposedly would have affected my DNA. But anyway, that's what this is about. So last chapter we talked about sickle cell. I told you that's going to be an example again. So here it is again. You're going to learn how mutations work because it's a mutation that causes sickle cell. This, I think, again, just vaccines. Oh, no. I think that picture was actually of insulin. So speaking of insulin, or basically DNA stuff that you learn in molecular biology, that's how insulin is made, is they take a gene that's human and they put it in a bacteria, and genes are just genes, right? So the bacteria doesn't know any better. It produces insulin, and that's where we get it from. We used to get it from pigs, but now we get it from genetically modified bacteria. So if you're afraid of GMO and you're diabetic, I don't know. Anyway, and I don't even remember what this was for. Obviously, that's a cow. I don't remember what your old textbook was getting at with that. Let's jump into it. Molecular biology. To me, this stuff is so fascinating. So the old textbook that we used to use, they started every chapter with like a relevant story, like something new, so you could relate that chapter to what's going on in real life. That book was published before COVID-19. Otherwise, that would probably be this story, right? COVID-19 would be the story. But at the time that this was published, that book was published, Zika was the big thing. But anyway, if you download the PowerPoint, you can read all about it if you want. I'm not going to ask you any questions. But again, the idea here is after this chapter, you should have a basic understanding and understand how viruses work. So, for example, I'm not going to teach you how COVID-19 works, how the virus, how that works. But with all the information I'm about to give you, if you were to look it up to read how the virus that causes COVID-19, to read how it actually works, how it infects the cell, what it does, you'll know what it's talking about, right? You'll be able to read it and understand it because you're getting your information, your basic information here. So the first thing we're talking about is DNA structure and replication. So I told you back in chapter 2, when we talked about biological molecules, we did talk about polynucleotides, right? We talked about DNA and RNA, and I told you it was going to come back. So here it is. It's coming back. So this part should be largely a review. But let's go through it anyway. Also, this PowerPoint, I've kept it from the old textbook. This is one of those chapters, again, where I think the old textbook taught it in a better order. So I'm sticking with the order from the old textbook. So if you're following along with my lecture with the new textbook, you're going to notice it's a little bit out of order. And some of my examples are different, but don't worry about that. And on that note, the old textbook kind of told this chapter like a story, sort of like they did with genetics. Remember, we talked about Mendel and what he did with the pea plants. And the same thing here. So even though I'm going to tell you this information as if it were a story, like a history story, you do not need to know any of these dates, right? So we're going to talk about this chronologically, but I'm not going to ask you anything about the dates. That being said, by the late 1800s, we did know that DNA was a part of cells, right? That's all we knew. We knew there was this stuff, DNA. We knew it was in the cells. That's it. Didn't know what it did. Didn't know what its shape was. Didn't know what it was made of. We just knew what was in there. By the 1930s, we also knew that there was one kind of molecule that was the basis of inheritance. So remember, Mendel already figured out all this stuff that we just talked about in this last chapter, right? All this inheritance stuff. Complete dominance, codominance, incomplete dominance, all that stuff we talked about. That was known, but no one knew how it happened. No one knew how it worked. But by the 30s, again, they knew that, okay, There's obviously some sort of molecule involved. We just don't know what the molecule is. Then in the 40s, they learned a little bit more about DNA. And again, still at this point, no one knew that DNA was the inheritance molecule or what it did. But they did figure out in the 40s that it's made up of DNA and protein. And you do need to know that. I've told you that before, right? So this is nothing new. We mentioned that earlier in the semester, and I think maybe even in the last chapter. No, when we talked about mitosis and meiosis, we talked about this, right? So here it is again. You need to know this. What are chromosomes made up of? DNA and protein. And we'll get more specific again, like we did earlier. We're going to talk about those proteins, but for now, let's just leave it at that. Any questions so far? All right. By the 50s, we had figured out the DNA was the molecule that acts as the hereditary material, right? So that was a big thing, right? To figure out finally in the 1950s. Imagine that. Prior to that, we all knew, or not we all, but scientists knew all about inheritance, like all that stuff that you just learned in the last chapter. But it wasn't until 1950 that someone finally figured out, oh yeah, it's DNA that's responsible for it. And speaking of which, and speaking of independent work, this is another good topic. Not just this, but anything I teach you, there's usually like, if you were taking a biology major, there's like a whole chapter on this. Like, so here we have one bullet point telling you that 1950s DNA was the hereditary molecule. I had weeks, maybe months learning about that. So for anything I teach you, I recommend or invite you to ask how. Like, question me. Question the textbook. How do we know that DNA is the hereditary material? I mean, I'm about to tell you. But for the other stuff, right? Like all the stuff I told you about photosynthesis and respiration. How do we know? So if you want to, look it up. Anyway, let's move forward. So far, as far as the exam is concerned, I haven't really told you much that's important. But we did get to this, leading us to what this whole chapter is about. Once we figured out the DNA was the hereditary material, that ushered in something called molecular biology, which is basically heredity, which is what we studied last chapter, except now we're really looking at it at the molecular level. So we're not just saying, all right, we're going to mate this thing with this thing and look at the offspring. Here, we're like looking inside cells. We're looking at the chromosomes. We're looking at the DNA. We're zooming in, if you will, and looking at the codes, right? So we're not just looking at the chromosome. We're literally looking, we're sequencing and seeing what the letters are in the DNA. Anyway, so that's the introduction. Let's move forward. The next word, attendance, I'm going to circle it. I'm not even going to say it. So if you're at home watching this online with your eyes closed, relaxing, I don't blame you. But open your eyes for a second. That's your word for attendance. Anyway, again, in the 50s, they figured out DNA was the molecule that's responsible for heredity. After that came the effort to figure out the structure. Because like I mentioned way back in chapter 1, this is the theme that we're going to see throughout this semester. And we have seen it, and we're going to continue to see it. This relationship between structure and function. So we didn't really know how DNA worked because we didn't even know the actual structure. And once we figured out the structure, then we could figure out how it works. To that point, at this point in the history lesson, if you will, they did know all the atoms and how they were bonded, right? So they knew how many carbons there were, how many phosphorus, and how many oxygens, all that stuff, right? They knew how many there were. They knew this one was single bonded to that one, and this one was double bonded to this one. They knew all that, but they didn't actually have the shape, right? The three-dimensional arrangement was not known. Again, it's the structure that gives it its function. Specifically, if we're talking about DNA, its function is to store genetic information, copy genetic information, and then pass that from generation to generation. So that is something you should know for the exam, is those three little bullet points there. There's not going to be specifically one question where I ask you that, but that's kind of, those three things will be things that will find their way into other larger questions. So you need to know what DNA is for, right? So thinking way back to chapter 2, we already talked about that, right? So you had to know that lipids were there for, like your fats are there to keep you warm and for cushion and for energy storage. And you needed to know that glycogen was the energy storage carbohydrate for animals, right? And now you need to know DNA stores genetic information, copies it, and passes it from generation to generation. And of course, that's nothing new. In a sense, those three bullet points sum up the last chapter, right? The last chapter was genetics. That basically sums it up. Of course, we went into more detail, but still. Any questions about this slide? Okay. Let's talk about the DNA and RNA structure. So that's what we've been hinting at. I keep saying once we figure out the structure, then we can figure out the function. It was a big deal to figure out the structure. All right, so this part's a review. As you already know, DNA and RNA are nucleic acids, right? So of the biological molecules, that was 1/4 of them, right? We had carbohydrates, lipids, proteins, and then finally nucleic acids, right? So here we are. This whole chapter is about nucleic acids. Well, sort of, not necessarily. Spoiler alert. Like you already know from last week's lab, DNA provides instructions on how to build proteins, right? So obviously that's going to be a part of this discussion. It's how we go from DNA to protein, but we'll talk about that later. Another thing that you already know, but you need to know again for this exam, DNA and RNA are polymers of nucleotide monomers. So just like back in chapter 2, when I said you need to know the polymer and the monomer, here we are again. You still need to know it. The polymer is called a polynucleotide. The monomer is called a nucleotide. When we're talking about DNA, so not RNA, when we're talking about DNA, there's 4 different versions of the monomers, right, of those nucleotides. There could be an A nucleotide, a C nucleotide, a T nucleotide, or a G nucleotide. Those are the 4 different types. Just like when we talked about proteins, and this is going to come up later again in this chapter. Remember, we said proteins, the polymer was called a polypeptide, and the monomer was called an amino acid. And remember, we said there was 20 different amino acids. So here we're saying for these polynucleotides, there's 4 different nucleotides, right? So that is definitely something you need to know. Any questions so far? Again, this should be a review. Even this picture is a review, but there's a picture, right? So there's your double helix. If we just take one of these strands and look at it, because remember, that's 2 strands, but we're just looking at one strand. And this is one way of looking at it, right? There's a nucleotide, there's a nucleotide, there's a nucleotide. All of those are nucleotides. This one on the right, it's the same thing, just a different way of drawing it, right? So there's a nucleotide, so on and so forth. That's all. We're looking at the same thing, just drawn different ways. Any questions so far? You notice too that there is a phosphate group and a sugar, but we're about to talk about that in an upcoming slide. All right, the nucleotides, how are they combined? How are they joined? This is another thing you need to know. This is very important. This is not trivial. It's almost trivial when we talk about proteins. It's not trivial here. They are bonded with covalent bonds. You need to know that. So let's see if anybody remembers way back from chemistry, because I said this was going to come back. I said you needed to know it later in the semester. Here it is. Is a covalent bond the strongest or the weakest of the bonds? Good, strongest. And that's going to be important later today, later in this lecture. Yes, very good. Nucleotides are the strongest of the bonds. So, covalent bonds are the strongest. So they are connected with the strongest of the bonds. Another thing you need to know is how they're connected. So you've got the phosphate of one group connected to the sugar of the other, right? You need to know that. So those bases, the A's, the C's, the T's, and the G's, they aren't involved in that. They're just kind of sticking out on the side. They're not involved in this. It's the sugar bonded to the phosphate, bonded to the sugar, bonded to the phosphate. Any questions about that? And I know this is boring. I love this stuff, and even to me, this is boring. But you have to understand this boring information to understand the other stuff I'm about to teach you, which is a little bit less boring. Again, the 3 parts. Actually, this slide should have come before the other one. My bad. Anyway, the 3 parts of the nucleotides. You need to know this: the nitrogenous base, the sugar, and the phosphate group. And if you're making flashcards, you could even just memorize base, sugar, and phosphate, right? Group doesn't really matter. Yes, it's a nitrogenous base, but for you guys, that doesn't matter. I'm not going to ask you that on the exam. So again, if you're just like making flashcards to study for the 3 parts of a nucleotide, sugar, base, phosphate, that's fine. You don't necessarily need to know nitrogenous or phosphate group. Also, the order that I wrote that down is completely random. So it doesn't have to be base, sugar, phosphate. It could be phosphate, sugar, base, or any of it. That's just randomly written. There is no particular order. that I wrote that. Any questions about that slide? All right. The nitrogenous base, like I said, that's the one part that kind of sticks off, right? So the sugar and the phosphate, we know those things are covalently bonded to each other. The nitrogenous base is not. It's not involved in that. It just kind of sticks out there. It does have a ring of nitrogen and carbon, but you don't need to know that. So let me put it next to that. I teach out of the textbook, so I included it. It is basic, hence the name nitrogenous base, but I'm not going to ask you that. And this part I've already told you, so you do need to know that. There's 4 different varieties, right? There's an A, a T, or C, or a G. Another thing about this that you should know, I should have written it up there. On that note, the idea that there's 4 varieties, that's part of what defines the nucleotide, right? Because I told you there's 4 different nucleotides. And that's kind of what defines it. So the sugar and the phosphate, those are 100% the same. If we're talking about DNA, an A, a T, a C, or a G, it doesn't matter. They have the same sugar, they have the same phosphate. But the part that differs, the part that defines it would be that. Let me go see what that's about. Excuse me. Make sure that's not important. Yep, nothing new. All right. Go back at it. Anyway, yes. So it's the nitrogenous base that defines it, if you will, right? That's the one part that's different. Everything else is the same. Any questions about the nitrogenous base? All right. Another thing that's not up there, but your textbook did mention it, so I'm going to mention it. I'm not going to test you. Some of these nitrogenous bases have single rings like this. Some of them have double rings. There's names for them. Your book talks about which one is which. So if you're taking notes, you can write down that I'm not going to ask you that. Maybe I'll ask you something like that for extra credit, but definitely not for a regular credit question. You don't need to know which ones are single rings and which ones are double rings. That doesn't matter to me. Anyway, let's talk about the sugar. It has 5 carbon atoms, as your book points out, but you don't need to know that. You certainly don't need to know that 4 of those carbons are in a ring and 1 is above the ring. And you definitely also don't need to know that the ring includes an oxygen atom. And you also don't need to know that carbons are numbered clockwise from 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. You don't need to know that. But your textbook mentions it, so I wanted to at least say it and tell you that you didn't need to know it. That way, when you're reading your textbook, you're not like, what? He never mentioned any of this. So, yes, that stuff's in there. Read it if you want. Learn it if you want, but I'm not going to test you on that. Maybe an extra credit question, but not a regular credit question. Just know that sugar is one of the 3 parts of a nucleotide. Anyway, any questions about that before we talk about the phosphate group? All right, phosphate group. As you might imagine, it has a phosphorus at the center. It is where we get the acid and nucleic acid because it is acidic. And it has a negative charge on one of the oxygen atoms. And I would just put a big old X through that as well. Because again, I'm teaching out of the textbook. I don't care that you know all that information. So in conclusion, for this whole big slide, you need to know the 3 parts of the nucleotides or nitrogenous base, sugar, and phosphate group. You need to know that the nitrogenous base is basically the one that defines the nucleotide because there's 4 different types of nitrogenous bases. You need to know the sugar and the phosphate are the same. They're not different between an A, a T, a C, or a G. And you need to know that sugar and phosphate are covalently bonded to each other, and the nitrogenous base has nothing to do with that covalent bond. That's the summation of everything on this big slide. So any questions about that? Okay. Whew. So like I already said, and I did make a slide for it, there's 2 different types. All right, so T and C are both single ring structures. A and G are both double ring structures. And there's names for that. But you don't need to know any of that. So put a big old X through this. The only reason I included it is because, again, when you're reading your textbook, I didn't want you to come across this and say, well, I don't remember him saying this. So I am saying it, but I'm also telling you I'm not going to test you on this. Again, maybe there'll be an extra credit question, but you do not need to know that T and C are single rings and A and G are double rings. Now, this is a little bit important, not for the exam, but I will point this out later. We're going to talk about how A's and T's always bond together, and C's and G's always bond together. And I'll go ahead and tell you now, this is one of the reasons why, because there's T's and A, right? So here's a T, and here's an A, right? So we have a double ring bonded with a single ring, and that makes the right width. If you had a double ring bonded with a double ring, it would be too wide. If you had a single ring bonded with a single ring, it would be too small. So that will come back later. I'm not going to require you to know that, but now you know why A's goes with T's and C's go with G, because it's the right size. Anyway, any questions about that side? None of that will be on the exam as a regular question, maybe an extra credit question. All right, the next word for attendance will be underlined right there. Anyway, RNA and DNA have the same chemical structure, except there are a few differences. There will be some questions on the exam about this. You should know the difference between RNA and DNA. First of all, you'll definitely need to know the difference between what they do. But before we get to that, this is the first thing you need to know is how they're different molecularly. First of all, when we talk about the different bases, right? DNA has A, C, T, and G, like we already mentioned. But now I'm telling you, RNA doesn't have a T. It has a U instead, right? So for RNA, the bases are A, U, C, and G. That is the most important as far as you guys are concerned. That will be important information later. But then also there's this, which is for you not very important, but we'll talk about it anyway. RNA has a different sugar than DNA. So for RNA, it's ribose. For DNA, it's deoxyribose, hence the different names. But I'm not going to test you on that. So again, for the exam, the biggest thing you need to know, well, one of the biggest things, there's another one I'll tell you about later. RNA does not have a U or T, it has a U. Which is also a good hint for the exam. When you get any question that has anything to do with RNA, if it's a multiple choice question and it has to do with RNA, if there's a T in the answer, then you know that's not the right answer. Because if it has a T, that means it's DNA. For example, and this will make sense a little bit later in this chapter, you're going to get a question where I say, here's the DNA strand, and I'll give you a bunch of letters, and I'll say, what is the RNA strand that's made from that DNA strand? And again, it's multiple choice, right? So when you're looking at all these different codes, if any of them have a T, you just know right away that that's DNA, and I'm asking for RNA, so you can, save your time and don't even read that option. You just know that option is not going to be one to correct. So any questions about this slide? We're making good time. If you download the PowerPoint, you can see this little animation that kind of talks about the differences and similarities between DNA and RNA. Same, if you download the PowerPoint, you can also watch a little video about this Hershey Chase experiment. I won't even get into it. You can look into it for independent work if you want. All right, here we go. Let's talk about the actual structure. So far, I told you about all the parts, right? We've talked about the nucleotides and the three parts of the nucleotides, but we even talked about how the sugar and the phosphate are covalently bonded to each other. But we still haven't really talked about the double helix itself for the shape. So let's get into that. It was James Watson and Francis Crick that are credited with determining the DNA as a double helix. It's not a history class, so I'm not going to ask you anything about Watson or Crick or Franklin, who produced an X-ray image of it, and they saw that, and they kind of copied off of her work. It's actually a little bit of controversy there if you want to look into that for independent work. So they got all the credit. She got none of the credit. There was also some other people involved too that didn't get much credit. But anyway, let's go back. Let me take a step back. What is a double helix? I'm not going to ask you the definition of a double helix, but I'm going to use that word a lot. So just so you know what it is, it's a spiral that has a uniform diameter, meaning, again, it's the same width. It doesn't get wider and then skinnier because, again, they have those same double ring bonded with a single ring, right? So it's always the same uniform diameter. And then, of course, it's twisted, right? That's what I mean by spiral, right? It's twisted. Anyway, like I said, they saw this X-ray from Rosalind Franklin. That was part of what helped them determine the structure. And using wire models, they tried to construct a double helix to conform to everything they knew about DNA. This, as far as the exam is concerned, this is a very unimportant slide, but I'm just telling the story. as it happened. Here's a picture of James Watson and Francis Crick on the left, Rosalind Franklin on the right. Actually, I'm trying to find, there's a video. I'm gonna try to find the video and share it with you guys. It's more like a movie. It's a decent movie because the story behind this, it's slightly interesting. Like I said, there's a little bit of double crossing going on there, a little bit of sneakiness. And what I like about these two is they were horrible students. They're always at the pub. They're drinking a lot. They didn't really work much. So it just so happens that those horrible students happen to be one of the most important people in biology by chance. But anyway, here we go. The model of DNA is like a ladder that's twisted into spirals, right? So you can see the picture down there. That one's not twisted, but that's what a ladder looks like, right? So the sides right here would represent the sugar phosphate backbones. The rungs in between would represent those nitrogenous bases. And then finally, here's some information I'm telling you for the first time, and you need to know this. The bonds that are keeping right here, these nucleotides together, right? The thing that's bonding this to this, those are hydrogen bonds. You need to know that. So again, sugar phosphate backbone, those are covalent bonds. It's keeping that single strand of DNA together, right? And it's covalent bonds that keep this other single strand of DNA together. It's hydrogen bonds that keep those two strands together. Does that make sense? And I wish you online could see me. Probably not because it's too zoomed in. I'll try to stand here. So one strand of DNA, that's a sugar phosphate backbone. Those are all covalently bonded to each other. This thing, this strand of DNA, covalently bonded to each other. But these two strands, when they come together, that's a hydrogen bond that's putting them together. And I would ask you this question, but we've already asked it. We already know that covalent bond is the strongest bond. Hydrogen bonds are the weakest. That's going to come back later. That'll be important later. So yes, it's hydrogen bonds that keep the two strands together. The weakest of the bonds. And speaking of that, there are base pairing rules that dictate which combinations of bases form the rungs. In other words, like I've already told you, and like you're going to see on an upcoming slide, A bonds with T, right? Those are always hydrogen bonded together, and C always bonds with G. Or vice versa, right? T bonds with A, G bonds with C, however you want to put it. The order is unimportant. The point is, those two are always bonded together. Those two are always bonding together. Hydrogen bonds. So any questions about that side? All right. Again, like we said, it's like a ladder that's been twisted. So there's a decent picture of that. Again, just some more pictures, some more visualizations. These are the same things, just drawn in different ways, right? There's a ribbon model of the double helix. There's the atomic model of the double helix in the middle. There's a computer model of the double helix on the right. Just an example. Oh, another thing, I don't remember if your textbook talks about it or not. I'm not going to ask you about it, but just so you know, I already told you one of the reasons, for example, that C bonds with G is because C is a single ring and G is a double ring, right? So that keeps it from being too fat or too skinny, right? We always want a double ring bonded with a single ring. The other reason that they always bond like that is because C and G hydrogen bond 3 times together, and A's and T's only bond twice. So those are the 2 reasons, chemically speaking, that C's always go with G's and A's always go with T, right? It wouldn't work any other way. Sometimes it does. There are mistakes. We'll talk about that later. But yeah, that's why it works that way. I'm not going to ask you that, but now you know. As a matter of fact, when we do work in this building, when we do stuff like where we're trying to sequence DNA or amplify DNA, one of the things we have to do is melt the DNA. We have to have it at a certain temperature so that those 2 strands come apart. And part of figuring out what temperature to put that machine at is figuring out how many C's and T's there are, or how many different nucleotides there are, because it takes more energy to break up the C's and G's than it does the A's and T's, because the C's and G's are triple bonded. Well, excuse me, have 3 hydrogen bonds. So yeah, that determines the temperature. Not that you need to know that. If you do it, if you download the PowerPoint, you can watch this little video, an animation about the DNA double helix. Here we go. Finally, in 1953, Watson and Crick published a paper proposing the molecular model for DNA. So all that stuff I just taught you about, they figured it out and published the paper in 1953. Not that I'm going to ask you that, but there you go. And I'm also not going to ask you this, but about 10 years after that, right, 9 years after that, they got the Nobel Prize for it. And as the old textbook points out, and maybe even your new textbook too, Franklin also deserved a share of that Nobel Prize, but she had died prior to 1962 of cancer. So she didn't get any piece of the prize. Anyway, any questions about that information? All right. So Watson and Crick wrote that the structure, again, they were the first people in the world to figure out the actual structure of DNA. And once they figured out the structure, their words, they said it immediately suggests a possible copying mechanism for the genetic material. So again, prior to this, not only did they not know the structure, but more importantly, they didn't know how it worked. But as soon as they figured out the structure, it just made sense to them. It's like, oh, I see how this works now. That's what we're about to talk about. And probably, I guess, next time we come back. But we'll go ahead and start that introduction. So DNA replication. We're going to talk about this very quickly. As you already know, you're a little bit familiar with this. DNA replication happens in the S phase of interphase, right? So if we're talking about the cell cycle, you know that. As you know, when the cell cycle reproduces, or when the cell reproduces, it has to duplicate the information and providing a copy to the new offspring and keeping one for itself, right? You already know that. That's old news when we talk about mitosis and meiosis. And like I said earlier, it's Watson and Crick's model that suggested how it works. And that's basically how it works. They knew, once they figured it out, it just made perfect sense. Like, all right, this bond is covalently bonded to itself. Excuse me, this strand is covalently bonded to itself. This strand is covalently bonded to itself. Those two strands are hydrogen bonded to themselves. So once they knew that, they just knew, like, clearly that strand, those two strands must open up because the code must be these letters, these four different varieties in the middle. So and that's basically how it works. When we come back on Wednesday, we'll finish that discussion. So I will either see you in class on Wednesday or at Roka's today. And your TA will be there in the lab if you choose to go to the lab today at 10. And that's it. Wait, let's do one last word for attendance. Save is the last word for attendance. Save. The what? Oh, yeah, the last lab? Yeah, can you send me an email to remind me? Okay. And I'll email it to you.